Thank you, Brenda. Good afternoon, everyone. My name is Kshitij Patkar. I am in Office of Pharmaceutical Quality, and me and my colleague, Dr. Jin Xu, today will present our talk. This talk is about assessment of extractable leachables information regarding manufacturing process equipment, as well as container closure system that is submitted in ANDAs. So this is the presentation outline. I will briefly introduce the topic of extractables and leachables. I'll talk about briefly what extractables and leachables are. Then I'll briefly talk about various safety concern thresholds that we typically use, an applicant should use to assess the safety of the leachables from equipment or container closure system. Then I will talk about how we assess extractable leachables information in ANDA for the manufacturing equipment, and Dr. Xu will talk about extractable leachables from container closure systems. So just to talk briefly about extractables and leachables. Extractables are the chemical compounds or entities that are released from the surface areas of either process equipment or containers into an extraction solvent under laboratory conditions. Typically, these extraction conditions are exaggerated conditions. That means they are a little harsher than the typical process parameters or process conditions, and that you typically have for any process like filtration, filling, what have you. Whereas leachables are the chemical entities that are released from the surfaces into the drug product under normal operations, like during the manufacturing process and over the shelf life of the drug product. A lot of good information about how to conduct successful extractable and leachable studies is provided in many publications, such as USB Chapter 1663 talks about how to conduct extractable studies. Chapter 1664 talks about design and how to conduct leachable studies. There are also technical documents available, PQRI publications available that talk about in detail extractable leachables from manufacturing equipment or container closure systems, safety control, safety thresholds, etc. So applicants should use all these publications when they are assessing their leachables in their drug products. These are some of the safety concern thresholds for different routes of administration that applicants should consider when they actually calculate analytical evaluation thresholds and assess the safety of the leachables. Most of the routes that are for the chronic treatment, the safety concern threshold is typically 1.5 micrograms per day. For the non-chronic ones, it's 5 micrograms per day. For the ophthalmic topical products, typical qualification threshold is 20 ppm. There are some routes of administration that are considered to be very high risk, such as epidural. And for those, the safety concern threshold could be even lower. And if applicants have any doubts about which safety concern threshold to use for such scenarios, they could contact agency via control correspondence. So I'll just briefly talk about how we assess extractable leachable information submitted in ANDA, particularly for manufacturing equipment. What we typically assess are the study protocols, analytical procedures, data. So we check if the studies are conducted for all the relevant surfaces that come in direct contact with the bulk solution. We also look at if extraction conditions are relevant, are appropriate for extractable studies. We also look at if worst case scenarios are typically considered for extractables as well as leachable studies. For the analytical procedures, we look at if the procedures are fit for purpose, if they are able to detect the potential extractables or leachables, if the reference standards used for these studies are adequate or appropriate, relevant. And then we also look for any method qualification reports provided for these studies, for these analytical procedures. We also expect to see data in that we see, we typically expect to see if the extractables and leachable compounds are individually identified and quantified. And this is the kind of information that's typically we see a lot of applicants do not provide. We also look at mitigation of extractables if they were found above analytical evaluation thresholds. And these mitigation could include engineering controls such as washing the filters or tubing with solvents or bulk solution, or if there are any leachable studies provided for the drug products for these extractables. For the leachable studies, if there are any compounds found above the AAT in the final drug product, then we look for how these compound risk is addressed, whether there is a safety assessment provided for these compounds, or whether applicant again has proposed any engineering controls. If the engineering controls are proposed, we would like to see a data to justify that these controls have worked. That means you may have to carry out leachable studies after the controls have been implemented. So for example, if you are proposing to wash your filters by say 1,000 liters, I mean just making it up, of say water or bulk solution, then we expect to see that leachable studies are carried out after again, and then now they show that there are leachables, those were above 80 before are now below 80, or they are not there or could not be detected. I'll also talk about this aspect of how the data is provided typically in the ANDA applications. So we observe that currently the extractable leachable information typically for the equipment is kind of scattered throughout different modules, and it's kind of tedious or difficult sometimes to even find the information. And sometimes the information could be missed if it is not easily accessible. So what we are recommending to the industry is that they provide a structured summary of the extractable leachables studies that they have carried out, in addition to the actual reports. That means the protocols and the data. This summary can be submitted in a structured format where typically you would use product attributes and the process variables next to the relevant, say, parameters that were considered for the studies. And I'll talk about how it can look like in the next slide. So this is an example of a structured summary. It can be an Excel sheet with different tabs, if you can see at the bottom. And you can see this, the tab number 1 could be the overview of the drug product with application ID number, what the drug substance is, what's the commercial strength, or the lowest strength for the commercial batch size, maximum daily dose, what's the pH, because all of these are very relevant to the actual studies. So if the formulation contains organic solvents, what organic solvents are there, how much, whether it's a chronic treatment or non-chronic treatment, which is useful to determine correct SCT. Then you could have the equipment tab where you have various columns. Column number A would be a description column. You can have the information about the process parameters. So what equipment is used, whether it's filter, tubing, bag, manufacturer, catalog number, surface area that's in contact with the bulk solution, maximum duration of contact, what the temperature for that particular unit operation is, So for example, filtration is carried out at, say, 4 degrees. That's very relevant because then the potential for leachables is lower if the filtration is carried out at low temperatures, and vice versa. Also, if there is any flushing that is proposed, and then along with that, you have extractable study report summary where what have you used as a surrogate for these process parameters in your extraction, and then largest extractable identified, its name, description, etc. And then similar information can be in the third tab for leachables. Again, you can answer some of these questions, or you can actually provide the numbers. So it's up to the applicant. But this is the type of structural summary that we are sort of envisioning, that if we could get it, our life will be a little easier. So what is the advantage of providing this summary of extractable leachables studies is that we can identify if there are any deficiencies in the studies right away. So then we don't have to go through the reports, and dig through the information. It will also reduce the amount of number of information requests for something that we couldn't find, but it may be there. And it's very useful for a data-driven evaluation of extractable leachable information from application to application. And we are recommending that this structured summary be provided in regional information section 3.2.R, and preferably in Excel format. So I have a question for the audience. In what module of the application the structured summary should be provided? Yep, you got it right. So thank you for paying attention, and Dr. Xu will present about extractable leachables from container chloride system.
Thank you, Chris. So let's switch gears to extractables and leachable from container closure system, CCS. Let's talk about the design of extractable study first. So before you do anything, you may want to talk to the manufacturers of the CCS. Ask them if they have performed any extractable study already on the CCS they are setting to you. And ask them what are the materials of constructions of the CCS, any information available from the literature, and look into your own lab to ask your own people if they have performed any extractable study on the same CCS or CCS that has similar components. The selection of solvents and extraction conditions is always very, very critical. It's one of the most critical components of the extractable study. And USP 1663 has a very good and detailed discussion on the selection of solvents and extraction conditions. And I'm not going to repeat here. You can always go to read that chapter. It is very informative. Just like the selection of solvents and extraction conditions, analytical method is another critical component of the extraction study, for the reason that you need a method to identify and quantifying the extractables. When the method is properly developed and qualified, the very same method could be used later on for extractable studies, which is the next topic, design of leachable studies. The study conditions are typically the same as the formal stability studies, meaning that you will put 3 batches on accelerated and long-term stability studies, and you'll test leachables on these 3 batches on multiple time points. And the reason to test multiple time points is to inform the trending of leachables. And you expect that towards the end of shelf life, the level of extractables will be level off. Just like extractable studies, analytical method is very, very critical. The method should be validated so that it is capable of identifying and quantifying the leachables above AET. The method validation is actually nowadays very standard according to USB 1225. You can go there and take a look. But there's one thing I want to bring to your attention is the limit of quantitation, LOQ, which is very, very critical. You want to validate LOQ to the level that is below AET so that you can quantify any leachables that are observed above AET. The calculation of AET is also published in USB 1664. It is derived from maximum daily dose and safety concern threshold, which Chris has presented to you. And if you are not clear about what MDD should be for your product and the assay you should use, you are welcome to submit a control correspondence to OGD for consultation. And I want to remind you that the inclusion of an uncertain factor is important. To account for the uncertainty of the analytical method, a factor of 0.5 multiplied by 0.5 or dividing by 2 is quite common. And this factor is actually dependent on the method. It could be different. Okay. When you see any leachables that are above AET, then you are required to identify the chemical structures as well as perform the toxicological assessment. And when you submit your application, you do need to submit both, not only the chemical structures, but also the toxicological assessment so that we can review your application. Another thing I want to bring your attention is the correlation between extractable and leachable. And this correlation is beneficial to you as the applicant of any application because this correlation allows for the control of leachables upstream at the QC stage of CCS or manufacturing equipment. For example, if you see the leachables are approaching the tox level towards the end of product shelf life, you know that risk is higher. In that case, one of the mitigation strategy is to control the CCS at the QC stage, meaning that you test the extractable of CCS when you receive a lot of CCS and determine and project from that correlation, you can project what the leachable level will be in the product and decide if you want to accept that lot of CCS for manufacturing or you want to reject it. Okay? The correlation will also inform post-approval changes. So it's a good stuff that when you want to make a change to your CCS, you can actually rely upon this correlation to project what would be the leachable of the CCS with the change. And that way you minimize the risk of the CCS with the changes. Developing a drug product based on a risk-based approach is always good to do. And extractable leachable evaluation is no exception. First, you want to look at prior knowledge. You may have already used the same CCS for similar product or different product. By similar product, I mean that you could have used the same CCS for the product of different strength, for example, and you might be able to use the data from that product to support the new product that you're developing with a different strength, given that, provided that the vehicle is the same. You may also use data from a different product packaged in the same CCS to support an application under development that use the same CCS, but the formulation could be different. In that case, you want to be careful though, because you want to compare the leaching propensity of the two different products, route of administration, and maximum daily dose of these two products. And the reason for that is that you do not want the new product to have a higher level of leachables compared to the one that has been developed and approved. There are some routes that are considered not as low risk. For example, oral route. When I say lower risk, it doesn't mean that it is no risk. We know that if we eat wrong stuff, we get into trouble. So the oral route still have risk, okay? You may able to develop a product without performing extractable leachable study for oral drug by referencing 21 CFR 174 to 186, the integrate food additive regulations. If the following conditions are met, Number one, materials of construction should be the same as those specified in the regulation. Use conditions should be the same as those, or the same or less stringent compared to those specified in the regulation. For example, some material can be used to package alcoholic beverages. And if you have a product that is a very simple aqueous solution or suspension, then you should be fine, because we know that your product will not be leaching more compounds from the CCS or equipment compared to alcoholic beverages. The CCS should also be tested according to the regulations using the same method, meeting the specifications specified in the regulation. And another point which is tricky is how do you compare a drug to a food? As an example I gave to you earlier, some materials can be used to package alcoholic beverage. And if your drug is a simple aqueous solution or suspension, then that is straightforward, knowing that your drug will not leach more compounds from the CCS compared to food. But if the drug is, but vice versa may not be okay. So just be careful when you compare a drug product to a food product. So time for a knowledge check. FDA published a guidance for industry, container closure system for packaging human drugs and biologics in A, 1989, B, 1999, C, 2009, and D, 2019. The correct answer is 1999. The year this guidance published isn't really that critical. What I want to bring to your attention is this guidance. This guidance was published 25 years ago, but this guidance, even today, still remains a valuable reference when you qualify your CCS for your drug product. With that, I would like to pass the podium to Derek Smith.